Hi Bio 30, lesson 2 for 17.1 today. I just want to take a quick peek at our last lesson, monohybrid crosses, with a quick review about the alleles that we were talking about when we talk about a certain gene for a trait, like the type of seed shape, for example. We could have two possible alleles, either a round or a wrinkled version of that gene, and one of them will be dominant. That means if you get one of each of those, the one that presents itself, in this case, the round would be considered dominant because with one of each, it will show up and present itself as a dominant trait. If you have two of the recessive, you would show up as a wrinkled trait. If you have two of the dominant, of course, it will end up being round, the dominant trait. Now, when we cross two individuals, we've got one here that we would call heterozygous because it's this parent, the male has a male plant, would carry a round gene from one parent and a wrinkled from the other, and that would mean that the gametes made, so if this was a plant, the pollen, the male pollen or sperm could have half of it would contain the dominant and half of it would contain the recessive because it's carrying one of each in its genotype. Whereas this female that we're crossing it with, this female got a recessive from both of her parents. So all of her egg cells would have the recessive in them because that's the only gametes that are possible. So if we're going to cross these two individuals, we're going to put them in the Punnett square. There's the male, and the two possible gametes are big R and little r. For the female, the two possible gametes are just two little r's. And when you do the cross, you can see what possible offspring we would get. So these that we crossed up here would be considered the parents. These offspring that they got would be their first set of kids, basically their first filial generation. And remember, in talking about these, they may ask you what physically, or the phenotypic ratio physically, what those offspring would look like. And you would have to say that they would be 1-to-1 ratio of round to wrinkled. Remember, here is our legend. Big R is round and little r is wrinkled. And if they asked you genotypically what they would be, it would also be half and half, 1-to-1 ratio. Half of them would be big R, little r, and half of them would be homozygous, little r, which would be wrinkled. So in this case, because physically we've got half and half, round to wrinkled, and genotypes, half and half round to wrinkled, these ratios end up being the same for these offspring. So that was a little review on monohybrid crosses. What I'd like to do is start off with looking at what you would have been doing in class, which is a genotype lab. This goes with your notes on page 13 and 14, where you figure out what your phenotype is, and genotype if you can. Your phenotype is for 9 different traits. So if you read through page 13 and 14, they go through and explain that which is dominant and which symbol to use for each trait. The first one, looking at earlobes, they say that the free earlobe, the one that hangs down here, the free earlobe is the dominant one. And for that, if you have that, your gene would be a dominant letter F for the free. If you have the recessive version that's sort of connected to the neck, you would symbolize that as a recessive F. So you guys can go through and check those all out, see what symbols they refer to them as. This next one, diastema, is if you have the, you don't have the diastema, so you don't have the space between your two front teeth, then that is dominant, and they used a letter M for that. But if you do have the space, they say that's recessive. And I just messed that one up. Okay, let me go back. Just looking at this, they say that the dominant is to have, if you have the middle diastema, space between your teeth, even if you got it corrected, but if you did have that, it was dominant, and the recessive is to have no space between your teeth. You guys can check those out. Your chart on that looks like this, and so you can check out the dominant and recessive that they tell you. There are symbols that they're using for earlobes. For diastema, they used M's. For the interlocking fingers, which the thumb is, which way the thumb lands, they used T's. And so you guys can fill these out. And I'm just going to show you for an example what I'd like you to do for earlobes. They want you to see which one you have. So if I, for example, have free earlobes, I can check in the data information that would be dominant. And so I would know using the letter F that my genotype is going to be, I don't know what's wrong with my pen right now, is going to be an F. However, I don't know what my second gene is, so you would just have to put a blank in there for the second gene. Okay? For the diastema, if you don't have that space, the no diastema, that is recessive, not to have the space. And if you, whatever you have recessive, and I'm just doing this for myself, you guys can fill these out for yourselves, and that will be a photograph of this data that you can take to submit as an assignment. But if you don't have the space between your teeth, you would be recessive, and you would know what your exact genotype is, because if you're recessive, you have homozygous recessive genes. It's just that if you have dominant, for example, I have brown eyes, so number 4, the pigmented iris, the brown eyes, is dominant when you have darker eyes. And if I'm dominant for something, I use the symbol, the dominant symbol for it, and I know that I have the dominant for my first gene, but I don't know if I have a dominant or recessive for my second gene. So when you are not sure, you put a blank for the second one. Okay, so I've given you some examples of how to work with these, and I'm hoping that you can read the information, check out your genes for your genes, your genotypes, and you know those by checking out what physically, how you present for each of these 9 traits, and then determining whether you're dominant or recessive, you can figure out your genotype. And so I look forward to hearing back from you on that assignment. Okay. I'm just going to move that up. Okay. So what we're going to do today is take a look at dihybrid crosses. And what that means is, let me just take a quick look over here to explain it to you, compared to monohybrid, where your individuals are just looking at one of the traits at one time. In a dihybrid cross, you are looking at 2 traits at once, and that's as complicated as we will get. But for example, we can look at round seeds versus wrinkled seeds, but also look at tall plants and short. And so we're looking at 2 different traits on that plant at the same time. So you, for example, could have the male plant be homozygous round, but be heterozygous tall short, and cross it with, in the female plant, maybe is heterozygous for the round, so that female plant has 1 gene from one parent that's dominant, 1 gene from one parent that's recessive, and they may be homozygous recessive and be short. So what that means is for each parent, you're looking at what their gene combination is, and you're trying to do a cross now to determine what kind of offspring they could have. This is a round, tall male crossed with a round, short female. Okay, how we're going to do this, I'm going to introduce you to this before we go through the notes. And basically, just like in monohybrid crosses where you had to figure out what gametes this individual had, they had either big R or a little r, dominant gene or recessive. This individual had only recessives, and you knew what to put into your Punnett square. It's a similar thing where you're just figuring out what the sperm cells for this male, what the egg cells for this female would have as combinations in them. So, for example, the R's, the round and wrinkled, those could be maybe on chromosome 6, and the tall short maybe on chromosome 10. So they are not found on the same chromosome, and they could mix and match. They could be found in any combination, which is why we've got to figure out what possible gametes they could have, by what combinations they could end up being in the gametes. So what we do with this guy is say, we take the first one and say that the big R, the dominant R, could end up with the dominant T. That dominant R, though, could also end up with the recessive T. And then, I'll just change colors here. Then we go to the second one and say the dominant R could go with a big T, but I see that I already have that one. So the dominant R could go with a little t, but I already have that combination. So those are the only two combinations that this plant's sperm could have. Now going to the female, we do the same thing. With the first one, the big R could be with little t, and we look at that one and we're like, we already have big R with little t, so we don't have to write it twice. Then we go to the little r and say little r could end up with little t, and the second possibility we already have. So those are the only possible gametes these two individuals would have. So when we put them in a Punnett square, we simply put the two possibilities for this male on one side of our Punnett square and the two possibilities for the female written together, because they will appear together in the gametes, and we put them in the other side of the Punnett square. Now we're going to do a similar thing as we did with our monohybrid crosses, and we're just careful that, remember with the parents here, we state what the R's are together. So we do the round genes together. If there's one dominant, one recessive, we always write the dominant first. But this first possible baby that they could have would be homozygous dominant R and heterozygous T. I'm just joining them together. And this possible baby over here, I put the dominant before the recessive here, and the big T, little t. So we've got, this one was round and tall, this one also round and tall. Over here, this one round and short, this one round and short. So when we have to determine what our offspring are, physically, usually with dihybrid crosses, they're only going to ask you physically what the babies look like, not the genotypic counting up, but physically we can see that round and tall is two of them. Two that are round and tall, and two of them that are round and short. And remember that we don't write it 2 and 2 to 2, so we always write it the lowest possible version of the ratio. So it's going to be a 1-to-1 ratio. Physically, phenotypic ratio is 1-to-1 of round tall and round short offspring for this cross. Okay, you can rewind this and do it step by step again, but we're going to be trying more of these, so you can stay tuned for more dihybrid crosses after our notes, which are very short because we just need to do some practice. Basically, where we're using two pairs of genes is a dihybrid cross, and segregation or separation of the alleles has no influence on the other, so we could get any combination, which is why we have to check out what all combinations are possible. So if you have one parent totally dominant for two of the genes and totally recessive for two of the genes, you know that this one, the only possible combination that could be found in the gametes for that one would be big T, big G. Only possible combination for this one would be little t, little g. So if you were to do that kind of a simple Punnett square, you could see that all the babies of this cross would be heterozygous 100%. It's a pretty easy cross to do because there's only one possible gamete for each one, and that would be our F1 first set of kids for these parents right here. What they're telling you after that is that what they may ask you to do next is do a F2, which is taking the offspring here and crossing them to each other. Now, when you have a cross like this, when you only have one offspring, sometimes they like to do that and say all the offspring are the same. So let's see what happens when we cross those with each other. And maybe you can see this already, but there will be 4 possible gametes here. If you're heterozygous for both of these traits, big T can go with big G can go with little t can go with big G, little t can go with little g. So there's 4 possible gametes for that individual, and the exact same for the other one as well, because they have the same exact genotype. Now, one thing you must do when you're making these gametes is keep the T's in front of the G's. So you notice when I made my gametes, my T's are always in front of my G's. And now we have 4 gametes for each of those individuals, which is why this will amount to a very large Punnett square that has 16 squares. And so what this is actually these 4 gametes that I made for each of these 2 parents that are listed right here, same ones. This is supposed to say big T, big G, little g. So these are exactly the same 2 parents that the cross is indicating above. And when you put your 4 gametes on each side, this is as big as our Punnett square will ever get, but we end up with a 16 square Punnett square. And you just carefully, if I was going to do like this one, for example, you're just carefully putting the T's together and the G's together. And if you have a dominant, put it before the recessive, always the dominant before the recessive. And you write your, the 4 letters of what that offspring would look like. And then your job would be to count it up. So remember, you're just looking at the physical phenotypic ratio, physically what these babies would look like, remembering what the key or legend had to say. So tall is big T, green is the big G, short is the little t, yellow is the little g. So I just start counting the ones that are the same. So tall green, there would be 1, 2, 3, 4, 5, 6, 7, 8, 9 that are tall and green. And so we've counted those up. There are 9 of them. Then we go to whichever one is next. This one is tall and yellow. So tall and yellow, we have 1, 2, 3 tall yellow. Go to the next one. It would be the short green ones. There are 3 of them. And then all we have left is 1 that ends up being short yellow, recessive. And so what you're going to see here is whenever you are crossing 2 parents that are heterozygous for both of their traits, heterozygous for both of their traits, for both of their parents, it's kind of a trick when you're doing crosses, but it will always be this ratio, this phenotypic ratio. It'll always be 9, 3, 3, 1. 9 of the dominant, 3 of the dominant and 1 recessive, 3 of the recessive and dominant of the other, and 1 recessive. So when you do questions coming up in the future, if your question is one that is heterozygous for both traits, for both parents, it is kind of a shortcut that you know it's going to end up being 93301. For all other crosses, though, we've just got to try them out and figure them out. So before we do more practice, we've got Mendel's second law of independent assortment that pops in here that says that alleles for one gene will separate, or that means segregate or separate, independently of the others when gametes are made. So how the T's separate has no bearing on how the G's separate, which is why we have to try out all combinations, because all combinations are possible in the gametes, and that's why we try the Punnett square. So they will always be independent assortment. They will assort independently of each other when gametes are made. Okay, this little question is asking you for these if there is a monohybrid gene combination for one trait. Remember the gametes here, if these are males, all gametes for this individual will have the dominant R, because that's the combination that they've got. There is no other possible genes that would find up in a gamete. If this individual is making sperm, half would have the dominant and half would have the recessive. Same with this, half would have the dominant, half would have the recessive. For this one, the combination is only that they have a dominant T, dominant R. Remember, we put those together when we're indicating gametes for dihybrid crosses. So for this one, remember the big T can go with big P. We have that one already, so we try the other one. Little t with big P. We don't need to write it twice. Two possible types of gametes for that one. For this one, you see that there are heterozygous for both traits, so we already know it's going to be 4 of them. So big R can go with big P, big R can go with little p, and I'll just change colors here so we can see what else I'm doing. Then I go to the second guy, little r with big P, little r with little p, and I'm putting a line over my little p's so we keep them separated from big P's, and it's a good trick. And so these now are the 4 possible gamete combinations if you have a parent that is heterozygous for both traits. So there's a dominant recessive for both traits. You know you're going to end up with 4 possible gametes. Okay, that's how you find out gametes. When you look at this page here, they're just looking at another example. It's kind of off a little bit, but that's another example of the 16 square Punnett square that I just did above, and showing you again another chance to try 16 square Punnett square and how you're going to end up with the same old 9-3-3-1 ratio. Both dominant will be 9 of them. Dominant for one, recessive for the other, recessive for one, dominant for the other, recessive, recessive. It will always be 9-3-3-1 for offspring if they are ones that are heterozygous for both parents for both traits. Okay, let's try a little bit of dihybrid practice here. If you guys want to stop the video and try this on your own, you are welcome to do that. And I'm going to just walk you through some of these so that you can be off and running on your own. This paragraph does give us the legend of what the traits are. They're using a big W for white and a little w for yellow. They're using a big D for disc and a little d for sphere. Now, always good to put that legend on the side anyways, even though it's in the paragraph, because you will refer back to that as you're figuring out what your offspring look like. So in this case, they're asking you about this individual that they describe. So if you read it, you will see that it is a homozygous white and a homozygous sphere is one of individuals, and the other individual is yellow and it's disc shape. So those are the 2 that we're crossing for number 1. So when I look at this parent, I see the only possibilities for gametes is to have the big W and little d together, When I look at this individual, the only possible combination of gametes is to have the little w and the big D together. So when I make the offspring for this one, remember, do the dominant first if there is, and dominant first for the D's if there is, and 100% of your babies for question 1 are going to be this genotype, which ends up being white and disc. So 100% white disc offspring. That's the answer. Okay, so you're just finding the parents that they're talking about, finding the gametes that are possible for those two parents, doing your cross. It just happens to be a really tiny Punnett square for this one, and just figuring out what this baby looks like. Now they're telling us to, no, this is F1, the first set of kids for this cross. Now they're asking us to do the F2. Now to do the F2, what you're doing is saying, I have to cross, that one that's heterozygous with another one of the same. It's taking these offspring and crossing them with each other. Now, I'm just going to tell you that we know the trick for that, so I'm not going to write out this particular one entirely, but you know that if it's heterozygous for both of the traits, for both of the parents, hope you guessed it already, it is going to be a 9-3-3-1 offspring. So 9 of them are going to be dominant, which is white and disc shape. 3 of them are going to be white and sphere. 3 of them are going to be yellow and disc. And 1 of them is going to be recessive, yellow, and sphere. Okay, so you guys don't have to take my word for it. You can try this cross out for sure and see that you get the 9-3-3-1, but that will be your answer. With the black mouse question here, when you check out what they're tell you for a legend, write it out anyways. Black is the big B, brown is the little b, and big T is for the short tail, and little t is for the long tail, and there is your legend to use for the cross. Number 3 is asking you about this cross right here. And so first thing we need to do is find out what gametes are possible. This one has 4 possible gametes. Big B can go with big T, big B can go with little t, and then you take your second one and say little b can go with big T, and little b can go with little t. So you've got 4 possible gametes for that parent. For this one, there is only, second parent over here, there's only 1 possible gamete combination. And so this makes a kind of a funny small Punnett square that conveniently fits right in my little space right here. So to do this cross, remember, you're going to put the B's first, dominant before recessive. When you do T's, put the B's first, recessive, big B, little b, and use a pencil because sometimes you make an error like I just did. And you are going to now, that you have the offspring, check out what this is. They're going to answer the question. It says, how many have black fur and long tails? And the black fur, remember you wrote a legend up here, is the big B, and the long tails are the little t's. And so you can see that to answer your question, it is these guys and these guys. So basically 50%. So your answer is 50% of their babies will have black fur and long tails. There you go. Number 4 is showing you what 2 parents you're working with right here. And first thing we do, of course, is it's the same, it's the same legend as the question before, so I'm not going to rewrite the legend. But for number 4, you want to get your gametes. This, of course, will have 4 gametes. I'll show you how that you do this again. Big B with big T, big B with little t. Then you go to the second one, little b with big T, little b with little t. So you're checking which two combinations the first one can have with the second genes and which combinations the second B can have with the other genes. That's one parent. For this parent, big B and big T, that you have already. This little b with big T, and you have that combination already. So this is all you need to do for this one. And you are putting the B's together, T's together, big B before little b, remember that. You must do that so you can quickly tell what offspring looks like. If you don't put the dominant one first, that will be very difficult to do. You'll make mistakes. So always the dominant before the recessive. That is the correct way to indicate your genotype. And once you've got it done, you check what the question is asking. They're asking how many are this exact one, big B, big T, little t. And when I go and check my cross out, I find that it's only one of my 8 that is the one they were looking for. So the answer is that 1/8 of them, and I guess you could put this in your calculator. If it was a numerical response, it might be 12.5% because 1 divided by 8 is that, 12.5. But 1 out of 1/8 is big B, big T, little t, as the question asked. Okay, for my last question on number 5, it's indicated right here that we had for the legend, they were talking about free or attached earlobes, and they told you free was a big E, attached was a little e, and then the other gene they talked about was widow's peak being dominant and straight hairline being recessive. So you can see I put a little line over the little w, but that's the legend that we're using for question 5. Go ahead and try it before you see the rest of this if you'd like. But basically in this section, I'm just showing you how I got gametes, how I got the 4 gametes for those parents. So the parents that they talked about were both heterozygous for both traits. So there was a big E, big W, and little w, and you guys know it that if you have for both of the traits, heterozygous genes, then you're going to have 4 gametes to work with. So big E goes with big W. Big E goes with little w. Go to the other side. Second guy. Little e goes with big W. Little e goes with little w. And of course, the other one is exactly the same. So it's got the same exact 4 gametes. You're going to take those 4 gametes and put them here. If this was one of these was a female, one of the male. In this case, all of the 4 gamete combinations are put the same on both sides because they both were the same genotype of parents. Now, you realize that you're always writing the E's first, so always writing the E's before the W's. Consistently keep doing that. Check that you're doing that carefully. You can see that I put my little line above the little W's just not to confuse them with the big W's because it will make sure that you never have an error. When you do this, then that is when you can check out the answer to your question. The question was, how many have widow's peak and attached earlobes? And you have to go back and check what that genotype would be. And the attached earlobes is little e's. So we're looking for little e's. And the widow's peak would be the dominant big W, no matter what was with it. So we're trying to find how many would be like that. And I can see that we have one here. I can see that we have one here. And I can see that we have one here. So the answer to the question is that there will be 3 out of 16 that will be widow's peak and attached earlobe to answer the question. But it also asks you how many will be males with widow's peak and attached earlobes. If they ask you how many are males with that answer, you simply say half of the babies statistically would be males. So the answer to this question is that you have to multiply that out, and it will be 3 out of 32. You can put that in your calculator. If it's numerical response, it would be 0.09, or just 3 out of 32 if you're just want to know how many. But that is the answer to this question. You figure it out. Remember that we already pegged it, that it was going to be a 9331. The widow's peak attached was one of the 3s, so I probably didn't really need to work it out because I knew it was going to be 3 of them, because they were asking about how many are one gene dominant and 1 recessive. It's going to be 3 of them. 9 of them will be dominant for both. 3, 1 dominant recessive, 1 recessive, 1 dominant, and recessive. So there's my trick repeated again. And that will be where we finish off today. I will give you guys some answer keys to trying out the questions